So it's my great pleasure to welcome Dr. Douay here today to give us a talk about his work on senescence as a mechanism of minimal residual disease and relapse in AML, and the ways to target that. It's clearly an incredibly important basic science and clinical question. And so it's a real thrill to have him come up to Boston to give this talk. Just very briefly, by way of introduction. So Dr. Douay trained in Germany, although barely an accent, very, very clearly very Americanized. And came here during graduate school, did part of his graduate school in California, and then did his postdoctoral training with Ari Melnick in New York. And for the last few years has had his own lab at Fox Chase Cancer Center. And I have had the incredible pleasure of collaborating with Dr. Douay on a project. He developed an incredibly useful co-culture system to be able to actually expand AML cells ex vivo, which was critical for work that my own lab was doing. So this is a particularly auspicious visit from my perspective. So thank you so much for coming, and I'm going to hand the microphone over to you. Thank you.
Hi, everyone. And first, I would like to thank Julie for the invitation and the kind introduction. And I also would like to thank everyone I had the opportunity to meet. So great interactions, and it's exciting visit for me. And so just to start this. So these are my disclosures. They have no direct relevance for this talk. And I like to start with a general overview. So we have a short introduction followed by a section on AML organoid model that we used for the following projects. So the next section is about research I did as a postdoctoral fellow in Ari Melnick's lab. And then the last section in part 3 is one research project from our lab, and it integrates certain aspects from the two previous studies. So before I move on, I would like to thank here at this point every collaborator involved in the first two projects. This is basically a great result of all involved and done and achieved by their contributions. And with that, we'll switch to our general introduction. So starting with the simplified classical model of hematopoiesis, we have at the top of this hierarchy these so-called the normal hematopoietic stem cells with self-renewal potential that differentiate to the different progenitor cells, which in turn then can differentiate into these fully effector cells that are needed for the various functions in the blood system. Now, mutation along this myeloid path can lead to malignant transformation, which in turn can result in acute myeloid leukemia, which is the most common acute leukemia in adults. And in the AML field, there is also this idea of a hierarchy, this cancer stem cell concept. At the top, we have these so-called leukemia stem cells with self-renewal potential that give rise to these partially differentiated leukemic progenitor cells and leukemic blasts that leads to this massive expansion of these cells, and that basically causes these detrimental effects in the human body. Now, many of these mutations are affecting epigenetic regulators. And one feature of AML or hallmark are the disruption of DNA methylation patterns. And just to give a brief introduction so that we are all on the same page. So here we see an unmethylated cytosine. This becomes methylated as the 5th carbon becomes 5-methylcytosine. DNMTs, DNA methyltransferases, catalyzes this process. And then this 5-MC can be further oxidized, called 5-hydroxymethylcytosine or 5-HMC. And this process is catalyzed by TET proteins, which actually can further oxidize this in the process of demethylating DNA, but that's not the topic of this talk. And mutations at this step, including those including those with IDH, basically interfere with this process, and one ends up with an accumulation of 5-MC. And at certain areas in the genome, preferentially at promoters and at enhancers, this is correlated with gene silencing. And the idea here is nothing novel. So this getting rid of this 5MC will activate again these tumor suppressor genes that are silenced in process of the tumor during tumorogenesis. So we here see the normal cells, and then here as an example the promoter sequence of cancer cells that can be then targeted, or it can be demethylated using these hypomethylating agents such as 5-ASA. They are also then called DNA methyltransferase inhibitors, DNMTI. And while 5-ASA can be used in combination with other drugs and shows efficacy, on its own in AML is not really showing us it has only moderate effects, even though it demethylates the DNA. The question then arises, why are these DNMTIs not really more effective? And this is how the first study started. And, not too surprising because the epigenome is multi-layered, and this is really old story. People have combined 5-ASA with various kind of histone-modifying enzymes, inhibitors like HDACs, and you name it. So the idea is then basically to target these various histone modifications and DNA modifications to further enhance the activation of the promoter. However, there is also recent studies have shown that tumor suppressors can be inactivated by loss of enhancer activating marks such as H3K4 mono and dimethylation, as well as H3K27 acetylation, specifically at the enhancers that can then lead to silencing of these tumor suppressor genes. And then I was wondering, how about actually targeting the promoter elements and the enhancer elements at the same time to further enhance the induction of these tumor suppressor genes, to restore the level or even go higher to just improve the therapeutic efficacy of these tumor suppressor genes. And at that time when I started this project, one factor was really received a lot of attention, that was LSD1 in the field of AML. And just to give a brief intro into the LSD1, it mediates inactivation of enhancers in hematological cells, in part by removing H3K4 mono and dimethylation. It has also, independent of that, it can also mediate silencing of enhancers through protein-protein interactions. And it has been shown that it sustains an oncogenic potential in AML, AF9, leukemia, and it's also required for the growth of various other subtypes like AML, ATOS, and so on. And it prevents differentiation. So just to illustrate here in a simplistic view, an active enhancer mark with its classical hallmark features of H3K4 mono and dimethylation and the H3K27 acetylation. This is just illustrating an inactive enhancer where LSD1 forms also a complex with HDACs that then leads to, removes these activating enhancer marks. And one can target LSD1 with specific inhibitors. We use this irreversible drug against LSD1. And the idea here was then, basically, or the hypothesis I had was, epigenetically silenced tumor suppressor genes in AML can occur through both aberrant promoter DNA methylation and LSD1 mediated enhancer effects. And the idea was then to do a dual targeting strategy. On one hand, we remove the DNA methylation at the promoters, and then also on the other hand, we target LSD1 to or to facilitate the formation of these enhancer promoter interactions. Now, going that route, I faced a few challenges here. One is we didn't know which genetically defined AML are actually responsive to this combination therapy. And the other point was that using cell lines for these kind of studies are not appropriate because they don't really reflect the epigenetic state of primary tumor cells, and nor do they really reflect the mutational spectrum. For example, there are much more TET2 mutant AML cases in patients. So there are more TET2 mutant primary samples than there are TET2 cell lines, mutant cell lines. And also, cell lines are usually less heterogeneous, and this is critical when it comes to drug screening purposes, because the more heterogeneous the disease is, the higher the risk there is drug resistance occurring or coming out of it. And but then on the other hand, using primary samples, the challenge is one can't maintain primary samples long enough in culture. One can somehow still work with it if it comes to cytotoxic drugs, you hit them while you take them into culture. But when it comes to epigenetic therapeutics, they actually need to proliferate, especially with 5-ASA that needs to be incorporated in the DNA. So one needs really happy, so to speak, happily growing primary cases ex vivo. And so what was the solution? Yeah, I had to spend a lot of effort and time in developing AML organoid models to see which one really is capable of propagating these primary samples. And while some models work better than others, at the end I decided to go with one that was efficient or more efficient in terms of time, cost, and reproducibility. And what I ended up doing is I used MSCs, here illustrated. They form these layers, and in culture with AML, what we see is, first of all, these are AML cells on top of the MSCs. We don't, it's so overcrowded, it's hard to see them. But then, interestingly, we can also see these flat-looking cells here in the center. What happens is these cells actually go under these MSCs, and they form these niches. And this is what you would expect in a normal bone marrow environment, that they form these niches. It is a key feature of the natural environment of the AMLs in the bone marrows. And because of that, we consider these as AML organoids. And they also maintain a polyclonal structure, and which we know is actually challenging when it comes to AML PDX models, because there is a greater selection pressure. One selects for certain clones with engraftment properties. Okay, before I come to this, I also want to mention here, a cornerstone of my lab's mission is to leverage these organoids to reduce the need for AML PDX models. And yeah, as Julie mentioned, this is how we got in touch, and when she told us she needed primary samples for a ChIP-seq experiment, but her only option at that time was to engraft a bunch of these IDH mutant AML cases in a number of mice with no guarantee that they will ever engraft, but with the guarantee that it will cost her lab tens of thousands of dollars. So, we were thrilled to collaborate with her. And yeah, we took a few of these IDH mutant samples in culture, expanded them, and things worked out. She got her cancer discovery paper accepted, and we were collaborators, so win-win. And one also could consider it as a win for the mice that they were not needed. And yeah, so, and I should also mention before I go, we are continuously improving these AML organoid models to further enhance their growth supportive potential. And we started also to build an AML initiative at Fox Chase. So for us, it really costs just a few $100s, and we are really happy to collaborate when you think, if you think this could be also useful for us. It's in its development, but we are moving forward with it. Now, moving forward, so the question here was, does this model actually recapitulate features one would expect to see in patients? That was very important for us, because because we want to use it as a surrogate to test or to recapitulate, or let's say it this way. We want to know if certain features that are present in patients can be recapitulated, so that will give us a decent preclinical model to study drug resistance. And to address that, I should mention we collaborated with Martin Carroll and Elizabeth Payeta, who provided us with hundreds of AML samples, and I found some that were derived from patients that had refractory disease or from patients that were poor prognosis. And then I decided, okay, let's take these guys, see how they respond to chemotherapy. So in AML, the main component of the induction therapy is cytarabine, also called RC. And what this box plot shows are the doses needed to kill 90% of the cells, EC90 doses. And what we observe is, even though there is a big spread, what one would expect for a heterogeneous disease like AML, there is a significant higher EC90 values required for these poor prognosis refractory cases. Hence, we recapitulate key features that occur in patients. So it is, as I mentioned, a decent preclinical model to also study drug resistance. Now, we leveraged this model for that one published paper in which we used close to 100 samples. They grew longer than 4 weeks, or at least 50 of them, more than 52 to be exact, grew longer than 4 weeks, long enough to do our epigenetic therapies or apply our epigenetic treatment regimen. I'm not going to go too much into detail, this is published. But one thing I want to mention, we specifically used the 5-ASA dose that did not induce DNA damage because at a certain dose, 5-ASA will act like a chemotherapeutic agent if it's too high concentrated. So that's why we used the dose that induced DNA demethylation, but didn't cause DNA damage. By the way, I forgot at the acknowledgement to thank my mentor, Ari Malik, so that just popped up in my mind. I should have mentioned that, so just to be specific here. Okay. And then these cases were then tested in, we basically assessed them 2 weeks later for their drug viability and, for their cell viability and cell growth using flow cytometry. And we also performed genomic profiling using panel sequencing, and then what I did is I basically integrated the information from the genomic profiling plus the cell growth, or basically the functional tests we got from flow cytometry. And what panned out was TET2 were the cases that were particularly susceptible to this combination therapy. And that's why I focused on these TET2 mutant cases and performed a series of functional experiments. What turned out, they are, we were able to reduce the leukemia repopulation potential, so they were susceptible to this combination therapy. And then I wanted to understand what is the underlying mechanism of this. And this is here just a Venn diagram for an RNA-seq performed on these TET2 mutant AML cells. And what we observe is, again, because we used a low dose of 5-ASA, we didn't see really much of effect of 5-ASA. LSD1 had a decent effect. We got around 700 genes induced. But then there were this slightly more than 50% additional genes that were induced that in combination of LSD1, of the LSD1 inhibitor with 5-ASA, that were not achieved by LSD1 alone. So I hypothesized that these cooperatively induced genes may or contribute to this enhanced therapeutic effect. And I was interested to understand where is this cooperativity coming from? So I performed ChIP-seq for various histone marks plus LSD1, ChIP-seq for LSD1, and integrated it with the RNA-seq data, and then analyzed the or performed gene set enrichment analysis in a certain configuration where LSD1 was bound just at the enhancer, and analyzed if these genes were cooperatively induced compared to just LSD1 alone. And what we see is, yes, these genes, when LSD1 was bound just at the enhancer, were cooperatively induced by combination therapy, while the LSD1 just bound at the promoter did not significantly induce these genes. So it told us, okay, LSD1 needs to be located at the enhancer. And then the next question was, okay, how about DNA methylation? So we use 5-ASA. What is 5-ASA doing in these cells? Now, to address that, we performed ERBS, which is a method that captures around 3 million CPG sites in the human genome, mainly or preferentially at CPG-rich regions. And what we observe is, and in genes that were induced by combination therapy at baseline, so we saw that they had at the transcription, or around the transcription start sites, DNA methylation that was then significantly reduced by combination therapy. And then on the other hand, we were wondering, okay, how does it look for genes that were not induced by combination therapy? And there we saw it was these genes had basically no DNA methylation. And then, how about LSD1 binding sites? Does this potentially also affect LSD1 binding sites? Or so does 5-ASA basically affect these sites? And to address that, so I looked at the DNA methylation levels at the LSD1 binding sites. And here what we see is basically LSD1 binding sites are not methylated. And this is also better illustrated in this coverage plot. We see that basically almost a mutually exclusive pattern at LSD1 binding sites. At the flanking sites, we saw again an increase of DNA methylation, and 5-ASA reduced that at the flanking site. So in other words, the genes that benefited from this combination therapy had LSD1 at unmethylated enhancers, and promoters became unmethylated by 5-ASA. And so, basically, making the long story short, we identified that GATA2 was really the one that induced strongly in this. And this is the locus of GATA2. We saw that the promoter was demethylated by combination therapy, and this is consistent with Ross Levine's and Ellen Shee's findings, in which they showed that loss of TET2 function in TET2 in a FLT3 ITD mouse leukemia model resulted in hypermethylation of GATA2 that could be then reversed by 5-ASA. And on the other hand, we see that LSD1 was bound at a distal GATA2 enhancer that has been previously annotated by groups including Ruth Devil. And what we observe is at this enhancer that H3K27 acetylation was stronger, or there were a greater abundance of H3K27 acetylation after combination therapy. And I reasoned, okay, this may suggest that there is a higher enhancer-promoter interaction, and to test that, I performed 3C, so chromatin conformation capture assay. And yes, we saw that the interaction between the GATA2 promoter and the enhancer were only significantly enhanced by the combination therapy. So the model was then that after combination therapy, we enhanced the interactions by removing DNA methylation and by inactivating LSD1. And we did functional assays targeting the GATA2 enhancer to really make sure that it is inducing GATA2. And yes, that's what we saw. If we used a CRISPR-I targeting this GATA2 enhancer, we saw that GATA2 was, the upregulation was prevented after combination therapy, and that also this enhancer was relevant for their survival. When we target this GATA2 enhancer, basically we saw a survival advantage. And then we found that actually loss of TET2 resulted in reduced levels of 5-HMC at the GATA2 enhancer. And if you remember this mutually exclusive pattern at the LSD1 distribution sites with the DNA methylation, so we hypothesized that maybe loss of TET2 results in a higher binding of LSD1, and I performed an ChIP-seq in HSCs that where I basically knocked down TET2 and saw, yes, after knockdown of TET2, we saw more LSD1 bound at this enhancer, and the enhancer had less of these enhancer activating marks. And looking by PCR, we saw that GATA2 was repressed in these TET2 deficient cells. And when we used or targeted then LSD1 with an inhibitor, we saw we could restore the GATA2 levels. So in summary, for this project, we proposed that TET2 mutant AML cells are particularly susceptible to combination therapy due to two aspects. The one, the previously described DNA hypermethylation phenotype, targeted by 5-ASA treatment, and then the novel finding that 5-HMC depleted enhancers after loss of TET2 functions are inactivated by LSD1 and can be reactivated by inhibition of LSD1. Now, I should mention, so these findings were decisive in initiating clinical trials in collaboration with industry, and still the great challenge with AML is, even though if they respond to treatment, there is a high chance they will relapse. And that's the heart of the second project, where I was really interested in understanding this relapse pattern. And this is just a simple illustration what happens in patients in the clinic. So at diagnosis, there is a high leukemia burden that after chemotherapy is reduced, the patient go into complete remission. But unfortunately, after a certain time, there is a risk of relapse, or there is a great chance of relapse as documented in these studies. And while this relapse mechanism is still largely unknown, one thing is certain, that the presence of residual cells left after treatment, often termed as measurable or minimal residual disease, is a strong independent marker for increased relapse and shorter survival in AML patients. And I was particularly interested in understanding these residual cells, what makes them so special to survive and then give rise to relapse. And yeah, so I leveraged the organoid model, used high doses of chemotherapy to really study the residual cells. And what I observed is, not too surprisingly, these cells stopped growing, went into senescence. Here shown using the classical senescence-associated beta-gal assay. But out of curiosity, I kept them in culture. What happens to these cells over time? And after some weeks, I saw under the microscope the cells started to grow again, and I was wondering, is it actually possible that these cells escape that state? And this is really how this project started. So first of all, as I mentioned, this is nothing new that they go into the senescence. And at that time, or let's say it this way, recent data suggest, or recent findings suggest that, yeah, it's actually, there is evidence that these cells may actually escape that. And when I started this project, I came across two papers during the early time of my postdoc that worked on p53 mutant cell line, and one in which they described cancer cells can escape oncogene-induced senescence. But these were all cancer cell lines, and I still wanted to know if really patient samples or primary samples are capable of escaping this state, especially in AML. So to go that, before I go that, before I move forward, I'll state the hypothesis I had at that time. So this is basically the model of the hypothesis I construed. So a senescent-like state can facilitate the survival to genotoxic exposure by allowing AML cells to survive in a latent state while retaining leukemia repopulation potential. In a nutshell, what it means is that the cells after chemotherapy go into this senescent-like state, survive at this nadir state, or at this nadir, and then give rise to relapse. Now, going that route, before we go that route, it's important to really explain the concept of cellular senescence so that we are all on the same page, because cellular senescence, can mean many things. And so starting really with the key findings described, or the landmark paper from Hayflick and Moorhead, in which they described, or in which they basically took normal cells in culture, expanded them, and observed that after a certain time, these cells lose their replication potential and stop growing, usually between 40 to 60 doubling times. And that's the concept of replicative senescence, describing a limited capacity to divide, or in other words, a replicative lifespan. Now, there is also the concept of premature senescence. You could consider it in a way of an early exhaustion in which stress, such as such as chemotherapy or radiation therapy, induce the senescence phenotype. Also, the oncogene-induced senescence would somehow fall into this category. And other cytokines and various other stressors can induce this premature senescence. Now, this shape of the curve, one can remind one of the idea how humans age. And, I'm not now sure if this has contributed to the idea, but the assumption is to induce somehow therapy-induced senescence in cancer cells to tip them over so that they come into the senescent-like state. So that this way we took care of the cancer cells. They are not going to grow anymore, and the danger is contained. However, I would like to point out that in the same work from Hayflick and Moorhead, they actually described that cells can acquire chromosomal aberrations and start growing indefinitely, becoming virtually immortal in the sense of their replicative lifespan. And unless one argues with religious figures like Noah or his grandfather Methuselah, I haven't heard about any human acquiring or getting close to immortality by acquiring or inheriting chromosomal aberrations or any other type of mutation. So where I'm heading with this is the following. Cancer cells have already acquired mechanisms to bypass replicative senescence. So the assumptions derived, or assumptions regarding senescence derived from normal cells do not equally apply to cancer cells. So that's, considering all this, it's not too far-fetched to suppose that cancer cells can also bypass or evade therapy-induced senescence. So going that route, though, one faces here also two challenges. One is cellular senescence is defined as an irreversible arrest of cell proliferation. So how do we solve this? Easily, we just call it senescence-like phenotype. So that's one challenge tackled. Now, the other challenge here is the technical one, that the classical assay of senescence fixes the cells. In other words, it kills the cells. And, it's a self-fulfilling prophecy. If your assay kills the cells, the senescence cells, it will become irreversible. So you basically, there is no argument that these cells will ever recover from that state. So the important question is what happens to these cells if one uses an assay that doesn't kill them and they remain viable? And so I came across this paper in which they used a fluorogenic or described a fluorogenic substrate called C12FDG that could be leveraged to detect and monitor these the senescence-associated marker in viable cells. And that was key for me and others. And also I should mention, other people also described this to bypass this issue. So, and just to illustrate that this assay or this C12FDG looks very similar in its form, how it manifests after chemotherapy treatment. And then I treated a bunch of AML specimens with this with chemotherapy and looked for the senescence marker, this fluorogenic senescence marker, and as you can see, they all upregulated the senescence marker. And I should mention, we tested also a few other chemotherapeutic agents, anthracyclines, that are also usually used in induction therapy, doxorubicin and daunorubicin. They also induce the senescence-like associated phenotype, but the problem with that is they have a fluorogenic property that would interfere with C12FDG, so that's why we used just RSC for this assay, for this fluorogenic assay. And, that marker, the senescence-associated beta-gal marker, is on its own not sufficient to really describe senescence. Other hallmarks are needed, or we looked at other hallmarks like increased cell size and granularity. We all observed that after chemotherapy treatment. And also, when we looked at transcriptional profiles, like SASP, senescence-associated secretory phenotype, or inflammatory response, we all, these gene sets or this transcriptome was induced after RSC treatment, and also other classical senescence-associated transcriptomes were upregulated. And obviously, we also looked at cell growth, which is then the last or the most critical part here, that these cells stop growing. And, I wanted to be sure that this is not just our observation, that this is, happening in other labs. So I downloaded Tim Lay's, published data from Tim Lay's group, in which they treated a series of AML specimens with RSC, also using a co-culture model. And what this box plot basically shows is the other normalized enrichment scores after RSC treatment, and anything above 0 means it's enriched. And basically what we observe is in almost all these cases, the senescence-associated transcriptomes were upregulated after RSC treatment. And interestingly, we also observed that diapause or embryonic diapause signature was induced in the senescent-like cells, and this was done in collaboration with Konstantin Mitiadis' lab and his lead author, Eugene Di Moloa. And at that time, we also actually did a triple co-submission with Catherine O'Brien and her lead author, Sumi Rehman. And how life goes, we all ended up in different journals. But it was interesting that we all observed this diapause or embryonic diapause-like signature in these cells. And and I should mention, we all use different cancer models, different chemotherapeutic agents. So it suggested there was some kind of a universally conserved stress response mechanism. And for those who are not familiar what embryonic diapause means, it's some form of a stress adaptive mechanism that mammalians can use where the embryo stops at an early stage during unfavorable conditions, like winter, for example, and then the embryo continues with its development during more favorable seasons like spring. Now, complementary, we saw that gene signatures that were associated, that were downregulated after diapause were also downregulated in these senescent-like cells. And yeah, genes associated with mitotic cell cycle, as expected, were also depleted. Now, it's obviously somehow related to a DNA damage response, and this will depend on the dose of the drug. So to better study this, what I did is I treated these cells with various doses of RSE and analyzed then the induction of the senescence marker. And at moderate doses or low doses, we didn't really see an induction of this senescence marker. But at moderate doses, 1,000 nanomolar is actually the dose that one approximately gets in the plasma concentration or has in the plasma in patients. And we saw at this dose that the senescence marker was induced. And at really high doses, there was a reversion of this. So, and this happened because the dose was so toxic that the cells, instead of going into senescence, they just died. And when we look at their cell growth over time, what we observe is these senescent-like cells stopped growing, but after a while, they started to repopulate the leukemia population. Now, the next question was, okay, can these cells also really engraft or give rise to leukemia in mice? So we saw that these senescent-like cells and engrafted them into mice and saw, yes, indeed, these senescent-like cells were able to engraft and give rise to leukemia. And interestingly, it actually engrafted better than the untreated cells, and this has been described by other groups, including Clement Schmidt, that there is some form of a senescence-associated stemness reprogramming. Once they arise from the senescent state, they become more tumorigenic. Now, really, we anticipated that derivatives or anyone could make an argument, you know what, you just enrich for leukemia stem cells. So to address that question, what I did is I sorted these senescent-like cells and performed single-cell RNA-seq, analyzed the data, projected different gene sets, and what we see is senescence, as expected, like SASP, was enriched in these chemotherapy-treated cells. Diapause was also occurred in a certain cluster in these cells, which is interesting actually. There is still some, it's probably not too surprising, there is still some heterogeneity even though we sorted for these cells by using the senescence marker. And then on the other hand, how about LSCs? We saw, depending on which signature one uses, but in general we didn't see an enrichment, so in some cases we even saw a depletion. So that means that enriching for LSCs wouldn't explain this higher engraftment. Now, the next question was, okay, does this really also occur in patients? So to address that, there were some published data from Mickey Bhatia's group in which they had patients with residual disease after induction therapy, analyzing these residual cells. What we observed again in 4 patients, in 3 out of the 4 patients, the senescence signature was up, and LSC signatures were consistently downregulated, consistent also with Mickey Bhatia's findings. And we also performed or analyzed published single-cell RNA-seq data. The same situation happened, that the cells and nadir had higher expression of these senescence-like signatures, and LSCs didn't occur, were depleted. And now looking at patients at relapse, what happened to these cells, or what are the transcriptional profiles when it comes to these cells? We observed actually there was a depletion of the senescence signature and an enrichment of the LSC signature. We did also functional experiment in PDX models to demonstrate that this is a stress response in which after chemotherapy the cells switch to a senescence-like program to survive, but then when they need to repopulate the disease, they switch to the stemness program. Now, to better understand the mechanism behind it, we looked for the classical markers P53, P16, and didn't see that or didn't observe that it's essential for this phenotype. Rather, we identified the NF-kappa B pathway and the ATR, and it's intuitive. It is a DNA damage agent that we add, and then it will respond. It will induce a DNA damage response. And I studied, or I tested a few other DNA damage mediators, but ATR appeared as the one that was most important for this. And this cartoon basically illustrates it. So chemotherapy induces a DNA damage, ATR becomes activated, induces the senescence SAS phenotype that promotes the cell survival. And if we then use an ATR inhibitor, we will basically bypass that. To test this, yes, we observe when we use an ATR inhibitor, the cells are abrogated to induce senescence or diminished, inducing this senescence marker. And the next question was, okay, does this really affect their viability? And yes, we observed after treating or after using the combination of ATR inhibitor plus the RSC, we saw in all 5 patient samples or primary patient-derived samples that the combination outperformed the single-agent drugs. And I should mention that this is not a this is not a synthetic lethargy approach. We actually blindside these cells before we add chemotherapy. So we prevent them to go into the senescent stress survival mode by first inhibiting ATR and then adding chemotherapy, RSC in this case. And serendipitously, other groups or another group has tested this in mice, in which they showed when ATR in combination with chemotherapy in an AML mouse PDX model improved their survival compared to the monotherapy. So that was all encouraging. And in collaboration with clinical investigators at Fox Chase, we are actively pursuing and are at an early stage to initiate a clinical trial in collaboration with Peter Abdelmessi and other colleagues. Now, to summarize part 2, so we observed that chemotherapy treatment induces in a dose-dependent manner a senescence-like state in AML. AML cells surviving chemotherapy can persist in a senescent-like state, and that this stress survival path or mechanism is mediated by ATR, and that the senescent-like AML cells can acquire stemness and repopulate leukemia in vitro and in vivo. Now, coming to the part 3, so we wanted really to understand how these senescent-like cells evolve, because we know that patients that relapse have less responsive disease, and patients also receive at least consolidation therapy after induction therapy, so several rounds of chemotherapy. And to model this and to see how these cells evolve, this project is led by Mana Ahmed, who is a postdoc in our lab. And what he did is he basically leveraged these organoid models that I developed and performed a single-cell clonal recurrence model in which he isolated cells, single cells, before RSC treatment and after RSC treatment at the first nadir treatment, or at the first nadir state, and then let these cells recover, and then isolated again single cells at the second nadir, and let these single cells grow and form a monoclonal population. So, in other words, an isogenic population. It's as close one can get for a genetically homogeneous population. And then the question was, how do these cells perform when we treat them again with chemotherapy, with the same exact RSC treatment? And what we observe is, not too surprisingly, the majority of these cells and parental cells died, and also the same in the first recovered clones and the same in the second recovered clones. However, we saw this gradual increase with each recovery cycle. So there is a gradual stress adaptation. And this observation, this pattern here, is, first of all, doesn't fit into the model of resistance, because if they really survived it through resistance, they would give rise to a resistant population. However, we saw that the majority of the cells still responded even after 2 rounds of RSC cycles. And then on the other hand, if we apply the drug-tolerant persister model, the idea here is that they reconstitute, again, a relatively sensitive population, so that also doesn't really fit into this. Now, one could argue, you know what, you're actually enriching for DTPs, rightly so. And in the world of AML, the closest that one can get to the DTP stage are the leukemia stem cells. And what we see is actually, yes, after the first round, we saw an enrichment, but after the second round, or in the second recovered parental clones, we did not see really a further enrichment that would fit into, that would explain this increased drug tolerance. And one could go crazy about it. So I should mention this CD34, which is the most established LSC marker. And one can really, what I want to say is, go crazy about these markers, but we thought, because of our previous findings, that it's something else. That this is more an epigenetic adaptation process, and what we observed is when we picked 6 different clones at each of these nadir states, and plus the parental cells, what we observe is that the parental clones cluster very closely each other, and then the first recovered population epigenetically drifted, and the second RSC recovered formed their own distinct epigenetic cluster. And we observed and I should mention this was done again using ERBS, and we saw gradually with each cycle greater DNA methylation changes, in particular in a hypermethylation phenotype. And when we really separate these different distribution of these DMCs, what we observe is it preferentially occurred at promoters, and we identified that GFI1 was the gene that was highly hypermethylated, and this was associated with downregulation of the gene. And when we look at TCGA, this is also consistent across the bank, that GFI1 methylation correlates inversely with GFI1 expression. And then the question was, okay, what happens if we downregulate or if we target GFI1? So we saw, after knocking down GFI1 in 2 independent patient samples, that they were had a survival advantage following RSC treatment. And the question was, okay, does this also have any relevance for patients? Yeah, we saw that patients with low levels of GFI1 have a poorer outcome. And some other groups have also mentioned this. So, and then when we look at the DNA methylation, so we observed actually the patients that have low levels of GFI1 had hypermethylation of GFI1. And then we wanted to know, okay, what happens if we really if we epigenetically target GFI1 by silencing them using a CRISPR-I construct? And yeah, here again, when we epigenetically silence GFI1, they have a survival advantage. And yeah, so what we observed is basically when we sought these nadir cells that survive treatment, these senescent-like cells, we observed that they epigenetically, undergo epigenetic variation. And this is by using an epipolymorphism algorithm that looks for the DNA methylation variability of adjacent CPG sites. So in summary, basically what we see is that these AML cells undergo epigenetic adaptation, and that GFI1 was one gene that was epigenetically silenced by promoter hypermethylation, and that suppression of GFI1 epigenetically and using knockdown approaches enhance the cell survival. And with that, basically, I conclude the talk, and I would like to thank everyone involved in this project. Thank you very much for the attention, and I'm happy to take any questions.